Hello ji, Namaskar. I'm Dr. Nutam Prakash Vishwakarma. And in last few videos, I have started discussing on the very important topic of bioinformatics, that was the sequence alignment. If you are new on this particular video, please go back in the playlist and watch the remaining the previous first three videos. Then it will be very easy for you to understand this video, the part 4. So, as I discussed in previous videos, what bioinformatics is all about, what the applications of it in different sectors, and three important criteria I have discussed in bioinformatics. The first one was the database, second one was algorithm, and third one is obviously the database. So, regarding the sequence alignment, I've also discussed about the dot matrix analysis. The dot matrix analysis had the demerit, or can say, so the demerit was like that. In dot matrix analysis, you can do the qualitative interpretation only by the just watching the graphics, but it have the limitation. If your sequence is too large, you will get difficulty to interpret the result, and only you can conclude something that whether two sequences are similar or not. And there was no explicit handling of gaps. You will not come to know that whether any gap is there or not. So then one more algorithm came, that was dynamic algorithm. The dynamic algorithm meaning very simple. If you have the very big problem, you can divide in the small sub problems, solve individually problems, and then club it up, and your alignment, you will get the alignment. And Needleman's algorithm was for global alignment, for pairwise global alignment. Now today I'm going to discuss about the Smith-Waterman algorithm, which we use for pairwise local alignment. So if you know the Needleman's algorithm, if you understand the algorithm of the Needleman, Smith-Waterman algorithm is very easy. But if you don't know the Needleman's algorithm, to understand the Smith-Waterman algorithm is going to be difficult for you. So what I suggest you, just go back and watch the part 3. In that one, I have discussed in detail about the Needleman's algorithm. So with certain modifications in the Needleman's algorithm, we can do the Smith-Waterman algorithm. So this particular algorithm was introduced by F. Smith and M. Waterman in 1981, and is the algorithm for comparison of sequence without aligning the entire length of the sequence. The partial sequence you can align. Say for example, you have a particular gene. You have newly isolated a gene. You identified a gene, and you want to know that what may be the function of this particular gene. So what you can do? You can go for alignment of your your gene sequence with the remaining sequences of the database. All right. So suppose you have genomes of many organisms. So once you go for global alignment between gene and genome, once we discussed about the Needleman's algorithm, in that one, we came to know that at the end of the alignment, you get the alignment and both the length of both the sequences become equal, right? So suppose your gene have only 10,000 bases long, and genome is 3.6 billion bases, million bases, right? Million, billion, whatever you guys. So there will be alignment, few alignment will be there, then will be thousands gaps or lakhs of gaps. In that case, you will not get any fruitful result, right? So in such cases, whenever there is a huge difference in the length of the two sequences, global alignment is not a good idea. Whenever there is the large gap, large differences in the length of the two sequences or more than two sequences, in that case, local alignment can give you the best alignment. Okay? So to perform a local alignment, we modify your global alignment algorithm, that was the Needleman's algorithm, by allowing a fourth option when filling the partial score table. As you remember, when we were filling the Needleman's algorithm, in that I told you that you have to compare among three scores, and whichever score is the largest, that you can put in your subset. But here one more thing come. There is a major difference. You have to compare among four scores. What are those scores? We'll see later on. And that score is 0. You have to introduce a new score from your side, that is 0. Let's see. So see, I have just I'm sharing with you the example so that along with me you also can perform. Meanwhile, you can pause the video and whatever I'm explaining you can write down in your book. You can solve. So see here I have two sequences. So just like Niedermann's algorithm, I have to put one row and one extra column, and then one extra column. Just like in Niedermann's algorithm, I told you that we are going to deal with the matrix initialization. So in the matrix initialization, what you have to do, what you did in the Niedermann's algorithm, you put it 0 and you keep adding the value of the gap penalty, right? So 0, -1, -2, -3. If it is gap penalty in your examination, it is giving -5. So you can write down 0, -5, -10, -15, right? Horizontally, and then left side also. But here is some change. In the Smith-Waterman algorithm, you don't have to add the value of gap penalty in 0 or in the subsequent classes, subsequent cells. You have to put only and only 0 in all the cells. This is the major change. So what I can do, I just put 0, 0, 0. So it's now easy, right? Because in the Needle-Manus algorithm, you might have write -1, -2, -3, so on and so forth. But here you have to put only and only 0. So it is going to be very easy. Now for the ease of doing what I am putting it, you can make with your pencil. You can use the pencil while you are solving the Needleman-Urus algorithm or Smith-Waterman algorithm. You can make no four temporary compartment you can make. Later on you can erase with the eraser, right? So four I made. And extra one, what I told you, no, the extra score means I told you that now you have to compare among four sequences. So extra you can put 0. Now three sub columns are there, now three subcells are there. Now what you can do, now you can go just upside. First make the practice, you can go upside. Once you will go upside, in the upside score, you can add the value of gap penalty, right? Just like the Smith-Waterman, Needle-Manus algorithm. So 0 and -1 will be how much? It is -1. Okay, all right. Now you have to move just left side. This make practice. Okay. So again, once you move in the left side, just left side, the value is 0. Add the value of gap penalty, and that you write down here. See, done. Now move in the diagonal. So once you go in the diagonal, how much you value get in this case? 0. And now in this diagonal's value, you have to add the value of either match or mismatch. So once I go just upside, here it just means outside the matrix. Here I got T, and here also I got T. So T and T is value is match. The match value is 1. So 0 plus 1 equal to 1. Now what you have to do? Now you have to compare among these four scores. So -1, 1, -1 and 0. So the largest score is 1. So that you have written. Alright. Now you can move with the next cell. Again, now you can erase all these values. Now what you can do? The temporary things I am putting. Now you can make the again the four sub cells. So that you can easily remember, no, you don't forget. Even if you don't write it, no problem. But while you are filling the values, you have to be very cautious, because if you commit a single mistake in a subcell, all the scores is going to be affected and your matrix will be wrong. So matrix, in the matrix filling, now what I can do, just go upside, because matrix initialization is over. Now we are doing the matrix filling. Now what you can do, just go upside. So again you got 0. So 0, in 0 what you have to add, you have to add the value of -1. That is 0 -1. See, an extra 0 I can put already. Okay, I can go just left side. So first what you have to do, first you have to make move to the just upside, then just left side here you can get 1, right? So z1 and gap penalty is how much? -1. So 1 minus 1, how much it is? 0. Now what you can do? You can go diagonal. So in the diagonal what I got? I got 0. So in 0 I have to add either the value of match or mismatch. Let's see whether I have to add the match or mismatch. So here you just go outside the matrix. Here it is C and here it is T. So C and T is what? It is mismatching. So mismatch value is 0. So 0 and 0, it is 0. Alright. Now you have to compare among all these 4 values. So the largest value is 0. The most beautiful part of this matrix is we will not get the value in the minus, because always you have to compare with this extra 0, right? So if any value comes in the minus, so automatically 0 will be larger than that. So you will not get any confusion in this matrix. You will get always either 0 or more than 0. All right. Now one more move. See, just make the practice with your pencil, right? And later you can erase it. Just go upside at the value, and before that I can put extra 0 here, so to avoid the confusion. All right. So just go upside at the value of gap penalty. So 0 minus 1 is how much? It is -1. Okay, then you go just left side. So 0 minus 1 is -1. Then you have to go just diagonal. So in that diagonal what you are getting? You are getting 0 here. Now C and T, it is mismatch. So mismatch value is 0. So 0 and 0 is 0. So again, how much value you get? Again you got largest value is 0. One more I am doing it. Few values I will do, then remaining you do. 4 subset. Just make the practice. What I can do? Just go upside at the value of gap penalty. So 0 minus 1 is 0 minus 1. Then you just go left side. 0 minus 1 is -1. And 0 you can put it here, right? Extra. 0 minus 1 -1. Then you have to go diagonal. In the diagonal 0 and A and T is mismatching. So value is 0. So 0 and 0 is 0. So again you can see here the largest value is 0. Very simple. Now what I can do? Again I can put the 4 subcells. So extra 0 I can put before. Just to go upside at the value of gap penalty. So 0 minus 1 is -1. Then left 0 minus 1 is -1. Then you go on diagonal. Diagonal value is again 0 at the value of either match or mismatch. So here it is A, here it is T, so obviously mismatch. So value of mismatch is 0. So 0 and 0 is 0. And the largest value is again 0. Alright. So you have filled the second row. Now next what I can do? Again the same thing. Now I have to go just upside. So value is 1. So I can, before I fill the value, just make the practice. I can divide in the 4 subcolumns. So 1 minus 1 will be 0 here, and the extra 0 should I can put on the 4th subcell. Then just go left side. So 0 minus 1 is how much? -1. Then I can go on diagonal. The diagonal's value is again 0. I can put the either value of match or mismatch. So T and T is matching. So 0 and 1 is 1. So here is the largest value you can see, that is 1. Alright. So likewise, you pause the video, try to fill all the matrix, then you play the video, right? So now you can match all your scores with my score given in the matrix. I'm not going to fill it because it is going to be lot of time. What you can match your scores with this particular scores of this matrix. All right. So now your matrix filling is over. Now we can move for the third and most important part, that is known as trace back. Okay. So in trace back, as you remember, while we were dealing with the Needleman-Wunsch algorithm, I told you that you have to always start from the right lower right corner from here only, right? and you have to reach finally up to 0. But here is the modification. What is the modification? In modification, you don't have to start from the lower right corner. Then what you have to do? You do one thing, you just try to find out the largest value in the matrix. Largest value in the matrix. So the largest value you can find out, the 3. So you have to start the trace back from this 3. Now, as you remember, I told you, you try to guess what was the origin point of this 3. So how to guess it? I told you that if it's this 2, this 3 came from this one, then what will be the value of? If it was originated from 1, so in the upper cell, what we did? We add the value of gap penalty. So gap penalty is -1. So 1 minus 1, it will be 0. Obviously, means this 3 is not originated from 1. If it was originated from 2, then it should be 2 minus 1, that is 1. So obviously this is not also the origin point of this 3. So what is left? The left part is 2. So obviously your origin point was 2. You can cross verify it. A and A is matching. So 2 in the diagonal value we added either match value or mismatch value. So match value is 1, 2 plus 1 equal to 3. Means you rightly tracked. Now let's try to find out the origin of this 2. If it was originated from 0, then its value should be -1. Means either, since we are comparing among the 4 values, so it should be 0, right? And if it comes from this one, it also again should be 0. So obviously this is not true in both the cases. So obviously the only part left is 1. So this was originated from this value 1. You can cross verify also. Here it is A and here it is A. As you know, in the diagonal value we can put the value of either match or mismatch. Here is the match. So 1 plus 1 equal to 2. Very simple. So this is came from here. Now what about this one? It should be -1 or 0. Obviously it should be again -1 or 0. So obviously it was this one was originated from C and C is matching. So 0 plus 1 equal to 1. Obviously true. See. Now, one more modification. While you are dealing with the Smith-Waterman algorithm, you have to stop putting the trace back whenever you get the origin at 0. You don't have to move from starting to end, right? You don't have to move. Wherever you get the 0 as a origin point, you have to stop your trace back. So your trace back is over. Now, what we can do? We can make the alignment. So in the alignment, you can see all the 3 diagonal arrows you got. So this is the base number. This is showing the base number 1. So from where you can start the alignment. Alignment you can put, see, one arrow represents one base. So here you can put A and downside you can put A. You don't have to include the T. Why you don't have to include? Because from here you got now the first arrow. So you have to start from here only. You have to start from here only. So what I can do? I can write down A upper side and so once your matrix filling is over, what you did? You just filled the trace back. Once your trace back is over, now this is the time for alignment. So in that, why do you are making the alignment? I told you you have to just watch the number of arrows. As much number of arrows you get, that much sequence bases are matching with each other. So only you can find out only 3 bases are matching in both the sequences. So what I can do? I can see the arrow, the diagonal arrow, right? So in the diagonal arrow, what I can do? You have to just start the alignment from wherever you see the no, uh, origin of the arrow. So means I have to make the alignment from this cell and you have to stop from this one, right? So in my alignment, I don't have to include this T because it is out of the range. So what I can do? One, as I already told you in the Needle-Manus algorithm, one arrow represent one base. So since the diagonal value is going, since the arrow is going in diagonally, so I don't have to make any changes. I can write down A in the upper side and this A in the lower side. Then see the arrow number 2. Again, one A you have to write down here, another A you have to write down from here. Then arrow number 3 you have to write down C and then write down C. So means only 3 bases are matching, C double A and C double A. And you can cross verify whether you have done it correctly or not, since you know that for each matches how much value we have to put 1. So there is 3 matches, so total score should be how much? It should be 3. And you can see this score 3 is matching with this one, means you have filled matrix very correctly. And if you want to make if you want to calculate how much percent of these two sequences are showing the similarity or identity, then simply you can see the local alignment, identity or similarity. Only 3 bases are matching out of 3. All the 3 sequences, because it is the local alignment, right? We don't have to consider all the sequences, all the bases. So there is total 3 matches. Only 3 out of 3 is matching. So 3 by 3 multiplied by 100 means equal to 100%. So there is the 100% identity between sequence A and sequence B locally, not globally. So that was all about the Smith-Waterman algorithm. Do some exercise, try to find out some questions, solve it by yourself, then only you will get the confidence to solve the Smith-Waterman algorithm. So we'll see you in next lecture and we'll start some another important session of the bioinformatics. I hope you enjoyed this session. Try to practice, then only you will be able to solve the local alignment and needle and global alignment problem. So if you like this video, please share and subscribe, and you can press the like button also. So thank you very much. See you in next session. Till then, take care. Jai Hind.